In this third screencast, we'll talk about excitation contraction coupling in the skeletal muscle. We'll spend a little bit of time on it, as I said, as it relates, so that we can compare and contrast it with what happens in cardiac and smooth muscle. We'll also talk about the sliding filament model. So a couple of diagrams here, just concentrate really on talking it through in steps. Step 1 is our acetylcholine release at the neuromuscular junction. Step 2, both of these are the neuronal side of it, if you like. Sodium enters through the acetylcholine receptor complex, as we've told you before, and initiates a muscle action potential. So that's the neural side of it, if you like. Down here you can see some important receptors that are involved. This is our T-tubule. This is something called the DHP, the dihydropyridine receptor. This is our terminal cisternae, arranged near the T-tubules, our calcium store, and this is how actin and myosin fit in. We'll see that there are two important proteins, tropomyosin and troponin. We'll talk about the role of these in excitation contraction coupling as we go through. So essentially this first slide summarizes the steps in the neural side of excitation contraction coupling, so the excitation side of it, if you like. Focusing in a little bit more on steps 3, 4, 5, 6 and 7. The action potential spreads down the T-tubules and alters the conformation of this protein here. This is the dihydropyridine receptor. If you're interested, it's called DHP, dihydropyridine receptors, because dihydropyridines are drugs that bind to the receptor, things like nifedipine, amlodipine, that you'll hear about in other parts of the course. The dihydropyridine receptor is a voltage gated calcium channel essentially, so the voltage alters the conformation, opens the calcium channel, calcium goes down its concentration gradient and triggers further release of calcium from these terminal cisternae. So overall, hopefully you can see that the action potential results in a large increase in intracellular calcium within the motor end plate. The calcium binds to one of these calcium binding proteins, troponin, allowing strong actin myosin binding. So binding to troponin is important. Again, we'll see maybe in a little bit more detail on the next few slides how this works and how it relates finally to the sliding filament model where actin and myosin cause contraction. The myosin heads execute a power stroke and the actin filaments slide towards the center of the sarcomere. So this is why the sarcomere is important. The sarcomere, remember, is the contractile unit between these Z lines, and it causes contraction. So let's consider the sliding filament model, the sliding filament theory for muscles in a little bit more detail. Contraction requires 6 components. There's a little mnemonic that you can remember, ACT 4. So A is for ATP, adenosine triphosphate, C is for calcium, and then 4 components beginning with T, that's the mnemonic ACT 4. So remember that ATP and calcium are important, hopefully you know that already. Thick filaments, thin filaments are important. Thick filaments are the myosin, thin filaments are the actin, and 2 other components beginning with T, tropomyosin and troponin. These are calcium binding proteins, and again we'll see how these fit in as we go through the next few slides. This is quite a lot of information. I'll go through it quickly and then I'll tell you the take-home message. This is the myosin, and it's really just showing you how myosin forms these thick filaments and what components, particularly this S1 component, are important contributors to muscle contraction via the sliding filament model. So myosin is a thick filament, has a globular head, and here's the head and tail region, present a sarcomere A band. Again, we told you what an A band was before. Essentially, it's the reason, or the reason we're telling you about these different bands is because they're important. It's important if you know how they fit into the sliding filament model. The tail of two myosin molecules wind around each other. That's one of the reasons that it looks thick compared to actin. Myosin can be cleaved by different enzymes, so chymotrypsin and papain. The take-home message really is that, you can see here chymotrypsin cleaves and papain cleaves further. So eventually myosin can be subdivided into these S1 and these S2 units, if you like. So again, take-home message, this is why I've highlighted it here. If you remember one thing from all of this information on the last two slides, it's the S1 fragment that's important for binding both ATP and actin. So take-home message, the reason that we're telling you about these cleavage sites and highlighting the S1 is present here is that it's this one that binds ATP and actin when we talk about the sliding filament in a little bit more detail over the next few slides. Similarly, lots of information probably about the thin filaments. We said that the thin filament is actin, and the binding proteins present on actin are tropomyosin and troponin, and this is just really a diagram to show you how these fit in. Present at sarcomere I band, the major thin filament is actin, and two chains are typically wound around each other, but they're still a thin filament compared to myosin. Importantly, each actin monomer, so that's the monomer, this is a dimer if you like, has a myosin binding site, an MBS, so shown here in this diagram, and these are important in the slide for the sliding filament model in muscle contraction. At rest, if you like, the actin myosin binding site is normally covered by the tropomyosin protein. During contraction, it's the troponin protein that's important for calcium binding to initiate muscle contraction. Calcium binding causes, or calcium binding troponin causes it to move off of the myosin binding site and initiates this sliding filament, which we're coming to shortly. Maybe again a little slide just really to probably stuff that we wouldn't really expect you to know in detail for an examination, but just to give you a little bit of extra information about where some of these Z lines and M lines come from. Thick filaments we know are made of myosin, thin filaments are made of actin. These are the take-home messages. It just shows you where some of these proteins are involved in a little bit more detail. Important take-home messages are probably shown highlighted in red here. So finally we come to the sliding filament model. Let's just take a little bit of time talking through this, because this is really the important part that we've been building up for. So hopefully we can see now that these are the this is the actin. We can see the myosin binding sites, and we can see the myosin here, and we can see why these myosin heads are important. And hopefully now we're going to see the importance of ATP and ADP and inorganic phosphate in this whole process. So let's start at number one. So let's say at starting position, myosin heads are attached to actin, and they form this cross bridge that we mentioned before. In this configuration, you've got ADP, adenosine diphosphate, and inorganic phosphate attached to the myosin head. The inorganic phosphate is released to initiate step 2. So in step 2, the myosin head pivots, as it says here, and bends and pulls the actin filament to start initiating contraction. ADP is lost and replaced by ATP. So this is the energy source. As the new ATP attaches to the myosin head, the link between the myosin and the actin weakens. So effectively the cross bridge detaches. So this is important. In this configuration number 1 and configuration number 2, actin and myosin are bound to each other, so the filaments can't slide across each other. ATP input removes this interaction and allows this sliding to take place, allows muscle contraction to occur. Step 4, ATP is split into ADP and inorganic phosphate. The myosin head is energized or cocked into the high energy configuration. And that configuration 4 allows the reattachment of the cross bridge. So hopefully that's nice and clear. You can see now why the myosin binding site is important. This diagram doesn't really show you the involvement of troponin, but troponin essentially, as we've seen on the previous slide, is the calcium binding protein that initiates the changes in the actin filaments that are important for the sliding filament. The changes to the myosin are initiated because the myosin head has a ATP binding site. When it's occupied by ADP and phosphate, the bridge is formed and the filaments can't slide. As phosphate and ADP is released and replaced by the energy source ATP, the bridge breaks down, the modules can slide over each other, the filaments can slide over each other and initiate contraction. The whole process then is cyclical, because ATP is hydrolyzed away and replaced again by ADP and inorganic phosphate. So we spent a little bit of time on that. Let's just then now finally just briefly consider how muscle contraction stops, cessation of muscle contraction. So as long as ATP and calcium are present, the muscle keeps contracting. Hopefully we can see that from the previous slide. Energy is depleted slowly, therefore we cannot suddenly stop contraction. There's another important protein called the SERCA protein. It's the calcium pump. SERCA stands for sarcoplasmic or endoplasmic reticular calcium ATPase. So it's an ATPase, so it breaks down ATP, very abundant protein. Around about 90% of the sarcoplasmic reticulum is made up of this pump. So a very important pump shown here. Calcium is pumped against its calcium gradient, so requires energy. So that's why it's an ATPase. So essentially, during muscle contraction, we get a large increase in intracellular calcium, as shown on those previous slides. If we want to stop muscle contraction, calcium must be pumped out of the motor end plate back into the stores, back into the SR stores, back into the terminal cisternae, using this protein SERCA. Action potential stops, no more calcium is released, but the calcium is high. So calcium binds SERCA, shown on these diagrams here. ATP is hydrolyzed and calcium is transported out of the motor end plate and muscle contraction stops. So hopefully this gives you an idea of the ATP and calcium. Remember that ACT mnemonic are two very important components for the sliding muscle, and the 4 other components beginning with T, thick filaments, thin filaments, tropomyosin and troponin are also important. So again, we won't spend too much time on this because we're coming towards the end of the allotted time. Skeletal muscle, why are they important for pharmacists? Why do we need to know about them? Because of these, obviously, for maintaining everyday life. But for a medical point of view, there are diseases of the muscle called myopathies, things that we'll learn about as we go through other parts of the course. So really just to mark your card that we'll talk about these as we go through later on, particular things like myasthenia gravis. I talk about those in other lectures. We have people at the University of Reading that are working on muscular dystrophy and also important ion channel diseases as well. So let's summarize here. We told you that skeletal muscle can be broadly divided into 2 main subtypes: red, slow, oxidative fibers, and white, sometimes called fast glycolytic fibers. Remember, there are intermediates. These relate to the type of muscle activity we want. We showed you that diagram of a runner. If you want to do long distance running, you probably utilize these muscle fibers. If you want short, sharp, dynamic sprinting action, you will utilize white fibers. Muscles, we told you about the organization. They're made of cylindrical muscle fibers containing myofibrils, made up of repeating sarcomeres. Hopefully you know the importance of a sarcomere now. They're separated by these lines and surrounded by the sarcoplasmic reticulum, which is important in excitation contraction coupling. This coupling involves coupling nerve inputs through the action potential and calcium and ATP dependent processes, particularly the sliding filament model, which you should be aware of, and why these cross bridges are important in allowing the sliding filament model to dictate muscle contraction. Okay, that finishes the screencast. We've covered skeletal muscle. In the next couple of screencasts, maybe 3 screencasts, we'll talk about cardiac muscle and smooth muscle, and maybe compare and contrast them with skeletal muscle.